at 11 o'clock. Good to see everybody. I have your exams graded, and I'll give you those back to look at today in laboratory. The scores weren't quite as high as the first midterm, which I guess kind of makes sense because the material is getting a little more complex now. But there was still a pretty good number of A's and B's, so give yourselves another collective pat on the back, though maybe not as vigorous a pat on the back as you did for the first one. Hopefully the third one will be a bit of a rebound and you'll do even better. Let's see what other announcements. I guess I told you on Monday that the quiz you'll take in here on Friday will cover this energy chapter that we're working on now. So that's what you should be studying for that. And I think we're probably going to finish the energy chapter today and start in on photosynthesis, but no photosynthesis questions will be on the Friday quiz. All right, well, let's just run away.
Is there an outline for our next exam?
An outline
Or when it'll be?
Well, it's in the syllabus, and
Okay, never mind.
Okay, you got me curious, so I'll look it up now. It looks like Monday, March 19th. Okay, let us see where we left off. The whole, or not the whole topic, but one of the major topics of this lecture was on how cells store up potential energy. And one way was in concentration gradients across membranes. But another method that cells use is something called making storing energy in what's called chemical potential energy, or high calorie molecules. And the basic idea behind this concept is this, that in the covalent bonds of molecules, there's potential energy stored in there. But there's a hierarchy, there's a ranking of what types of covalent bonds have more potential energy stored in them. And if we focus on the bonds connected to carbon atoms, the carbon-oxygen bond is the lowest potential energy type of bond that a carbon atom can have. A carbon-carbon covalent bond is a little bit higher potential energy, and a carbon-hydrogen bond is very high potential energy. So one thing that cells can do to store potential energy is to take low energy molecules like these ones, because all their bonds are carbon-oxygen, and break some of those bonds and convert them into higher energy carbon-carbon and carbon-hydrogen. So now you have potential energy stored up, as you can see from the potential energy diagram. And I guess I don't have the overhead for this now, but a good example of this is photosynthesis. When plants are doing photosynthesis, they're taking carbon dioxide out of the air and converting it into glucose carbohydrate. And why they're doing that is a way of storing up energy for themselves. Now, at some point, the cells need to tap into that potential energy that they stored up in the high-calorie molecules. And what they do in that case is essentially run the process in reverse, like this. They take high-energy molecules like glucose and then break those bonds, those higher energy bonds, and convert them into the lower energy carbon-oxygen bonds. And so this is a spontaneous exergonic reaction. The carbon atoms are losing energy, but the energy that the carbon atoms lose as they go down the potential energy diagram can be utilized by the cell to stoke up its own energy, the cell's own energy, namely in making ATP. And this process, which we're going to study in a week or two, is called cellular respiration, where the cells take glucose, convert it to carbon dioxide, and use the energy that the glucose loses to recharge their ATP supplies. Okay, and I think the actual spot we left off on was about a concept called activation energy. When I showed you this diagram, I said, all right, this is a spontaneous reaction. It's a downhill reaction, and all substances in the universe want to lower their potential energy, just kind of a law of the universe. And if this were water, of course the water would just rush down the hill automatically, but the glucose will not automatically turn into carbon dioxide. If you think about that, all of these common food items, for example, are made out of starch, which is just polymerized glucose, but they don't go poof, we just turn into carbon dioxide gas. So why doesn't that happen if it's a downhill reaction? Well, the answer is something called the potential energy, the activation energy barrier, which on the potential energy diagram looks like this. So it's defined as the energy that you need to put into a spontaneous reaction to make that reaction occur. In other words, if you have some glucose, you have to put some energy into it before it will go down and turn into carbon dioxide. The reason why there's this activation energy is simply that you have to break these covalent bonds before you can turn them into carbon-oxygen bonds. And that takes energy to break covalent bonds. Now, if you wanted to get this to happen, if you wanted to turn glucose into carbon dioxide, you can do it by adding heat energy. If you get, I don't know, some starch or some sugar or something and put a flame to it, then you provide so much kinetic energy to the molecules, they eventually bang into each other hard enough that they get enough energy to go over the activation energy barrier. Their kinetic energy drives them over there and their bonds start breaking. But that process is not available to a cell. A cell cannot have a little flame inside of itself to cause its molecules to get over the activation energy barrier. So how do cells carry out spontaneous reactions like this? Well, the answer is the enzymes, which we talked about previously. They are proteins that carry out chemical reactions, and they have that active site, that little crevice in the side, and the enzymes grab their substrate molecules, the molecules they work with, the substrates fit into the active site and get changed into the product molecules. So when I was showing you this before, I just said enzymes are proteins that carry out chemical reactions. But to be a little bit more specific, to use the proper terminology, enzymes are catalysts that make reactions occur by lowering the activation energy. When an enzyme binds its substrate molecules and brings them together in the active site, the act of the enzyme bringing the substrate molecules into alignment and actually pushing them together slightly lowers the activation energy of that reaction. So this is a spontaneous reaction, changing these molecules into this molecule. But if this was inside your cells, it wouldn't go because there's a large activation energy required. But if there's an enzyme to carry out that reaction, then the enzyme essentially reduces the activation energy. It shaves off the hump on the potential energy diagram. And so with the enzyme present, this reaction would proceed inside you. And so for all chemical reactions that take place inside cells, there are always enzymes that are the catalysts. And catalysts are substances like this that speed up a chemical reaction by lowering the activation energy, but the catalyst itself is not consumed in the reaction. Now, here's a diagram from your textbook. Actually, it's from a former textbook I used to use, but it's showing the same thing, that the enzymes lower the activation energy is what it's saying. Now, here's a non-spontaneous reaction, because this molecule is gaining potential energy as it goes from there to there. This hump right here is still considered the activation energy. And I think I posed this question last time. If there's an enzyme to carry out this process, does it become a spontaneous reaction? And the answer was no. Because as long as the products have higher potential energy than the substrate, it's always going to be a non-spontaneous reaction, and an enzyme can't change that. All an enzyme can do is lower the activation energy hump slightly, but an enzyme cannot make a non-spontaneous reaction spontaneous. In other words, the cell is still going to need to have some sort of energy input into this reaction to make it occur. Now, the enzyme might lower the amount of energy that you need to input by lowering the activation energy, but still you're going to need to have energy coming into the reaction to make it occur. And well, typically, as you've heard me say probably dozens of times so far, ATP is the energy source that the enzymes use to carry out non-spontaneous reactions. Yes.
So to lower the energy activation energy, is it somehow taking away, or is it adding energy to it?
I guess the actual physical chemistry of it I don't fully understand, but if I had to sort of explain it from a non-technical standpoint, imagine this is a molecule and this is a molecule. If you want them to react with each other, they have to hit hard enough to break their covalent bonds. And the chances of them hitting just right and just the right orientation is not so good. But if you have an enzyme grabbing them and guiding them to the right orientation, that reduces the amount of kinetic energy they have to carry out that reaction. That's the way I think of it. All right, well, just to review, enzymes speed up spontaneous reactions by just lowering the activation energy, and enzymes will help out non-spontaneous reactions also by lowering the activation energy, but you still have to put energy into a non-spontaneous reaction for it to occur. All right, remember that the cells are full of thousands of organic molecules just drifting around inside the cytoplasm, and the word metabolism means all the thousands of different chemical reactions that need to occur to keep the cell alive and functioning, and that enzymes are the catalysts that carry out all these thousands of chemical reactions. And let's just use these molecules as examples of some of the organic molecules that are drifting around inside the cell. Maybe from their names or their structures, you might remember what types of molecules these are. Does anybody recall? They're monomers of some type of molecule that we talked about.